So now we want to talk about the three divisions of the vertebrate brain. And so from the embryonic neural tube that develops in vertebrates, which that notochord develops into the nerve tube in development. And the notochord is something that's found in all chordates, but in there's one group that doesn't have, that's not a vertebrate, that's the amphioxus, which it doesn't develop into a nerve cord. It doesn't develop into the brain. But this consists of the neural tube develops into these three parts: the hindbrain, the midbrain, and the forebrain. And so there's two divisions of the hindbrain, the metencephalon, the myelencephalon, the mesencephalon. Mes means middle, and myelin, you know, that would mean that it has like white matter. It has a myelin sheath around it. And just whenever you see encephalon here, that just means brain. And so, you know, just know that that part means brain. And all of the precursor parts mean something, like this one means middle. And telencephalon means like transmitting to distances, like a telephone or a telegraph or something like that. And so you have the telencephalon here. And then the diencephalon. So it used to be considered just two parts, and now we actually know that it consists of three parts. So we'll talk about each of these different parts. And so in the development stages, these are what they're called: met and myelencephalon. The midbrain is called the mes, and then the forebrain is called the telen and diencephalon. But each of these components becomes something. And so let's look at kind of what they become. So the metencephalon becomes the pons and the cerebellum in the brain. And so, you know, it's good that we did the nervous system in our first anatomy lab, so you kind of know what we're talking about here. And then the medulla oblongata. Oops. And then in the midbrain, there's actually a couple of parts to this, which your book doesn't really talk about. I'll go ahead and mention them. They're the cerebral peduncle and the corpora. Oops. I'm using a new stylus here, so it's kind of different. So, corpora quadrigemina. And so the quad part here, that is what means four, and Gemini means paired, and corpora means body. And so on one side you have, of the midbrain, there's just a bunch of like tracks running over like this in the middle part. And on the other side it has four little circle areas called the corpora quadrigemina. And they have some really important functions in respiratory rate and heart rate. And so just saying that is important. They are important structures, which we learned about in A&P, but for the purposes of this class, just know midbrain is metencephalon. And then, or mesencephalon. And then the forebrain is the telencephalon and the diencephalon. So this is the cerebrum. And that is like what we usually think of just as the brain, proper brain, where all of the functions you normally think of, where you learn and store memories, that would be in the cerebrum. And you have your cerebral cortex and all that make up that cerebrum. So then the other part is divided into, really, it used to be two parts, and that's why it was called but there's really three parts. And so this is the hypothalamus, and the thalamus, and the epithalamus. And so those three sections are making up the part that we call the diencephalon in the brain. So here you can see in red, that's the whole kind of midbrain area right there. And then this area down here, this is the hindbrain. And then the area in blue, that is all considered the forebrain. And the parts, you know, they show the cerebrum and then the thalamus up here and the hypothalamus, which is down here, and then the epithalamus, which is not really shown in this particular view. The midbrain, like I circled in red, and then the rest of this, you know, like your pons, and then the medulla oblongata down here, and then the cerebellum make up the, so the pons and the cerebellum make up the met, and then the myelin encephalon is the medulla oblongata. So these are the developmental stages of like the embryo brain, but we don't call them that in the adult brain. You could think of them, I still think these divisions are pretty valid, but they aren't technically there. They develop into those parts in the adult brain. So evolution of increased brain complexity. So the size of its forebrain and its major subdivisions increases in animals with more complex nervous systems. So the complexity is based on the size, and it also has an increased surface area, and the convolutions, which are called gyrus and sulcus. So the gyrus is what is a groove, and the sulcus is kind of a fold. So these are the folds of the brain. When they say convolutions, that's what they're talking about. So by the fold, we talked about this fact that surface area increases when you have folds, and in the brain that is extremely important. So the more folds, the more surface area, and the more brain capacity. And so then we have you can see some of the different brains, and so the rat compared to the human is obviously smaller. As it gets bigger, you can see the cat starts to begin to have like folds in it, and cats are really smart. They know more than they think we think they know. They understand everything we're saying. They just choose to ignore what we say. And the sheep, although they are pretty dumb animals, they'll just follow anybody. They have more complexity in their brain, and they have more folds and larger size. Dolphins, though, look at how big the dolphin's brain is and how convoluted it is compared to the chimpanzee. So dolphins have long been thought to be very intelligent animals, and we don't know much about them as much as we would like to know, but they're learning more. So you can see the relationship between the body size and the brain mass here with this little, with the exception of the human, the dolphins generally, the greater the size and folding provides more surface area and more processing area and more interpretation of information. And so generally the body mass and the brain size are proportional, but humans and dolphins have a much greater proportion, compared to their body size, their body mass. So structure and function of nervous systems in humans and other vertebrates. The central nervous system and the peripheral nervous system are anatomically and functionally connected. The central nervous system receives information from the peripheral nervous system, and the central nervous system interprets that information and may initiate a response. And then the response is carried out by the signal that's sent out to the peripheral nervous system. And so you can see the peripheral nervous system, again, we've discussed this before, consists of nerves. So you can see the cranial nerves and the spinal nerves and the ganglia. Those are all part of the peripheral nervous system. And then the central nervous system is consisting of the brain and the spinal cord. So some terms that you need to understand about the vertebrate brain before we go too much further. These two terms are linked. They mean the same thing. They're a collection of cell bodies of neurons that are, but the difference is if it's in the central nervous system, we call it a nucleus or nuclei. And if it's in the peripheral nervous system, it's a ganglion or a ganglion. And then these two terms are related as well. The myelinated axons are in the central nervous system. We call that a tract. So in the brain and the spinal cord, it's a tract. And when it's in the peripheral nervous system, we call it a nerve. And then the cranial and the spinal nerves are connected in the way that they are nerves, but the nerves that are connected to the brain are cranial nerves, and the spinal nerves are the connection between the peripheral nervous system and the spinal cord. So they are coming out of the spinal cord, and so the name makes a lot of sense. And the different types of matter. So you've probably heard as your brain is gray matter. Gray matter has a technical term, and it is the neuronal cell bodies and dendrites and unmyelinated axons. So if there are axons in there, this is all unmyelinated, and that is why it is gray, and it is darker in color. Gray matter is not myelinated. White matter is myelinated, and the myelinated tissues are usually the axons, and they form together to make tracts or nerves. So the types of matter, you have white matter, which is myelinated axons, and gray matter, which are neuronal cell bodies and dendrites and axons, but they're all unmyelinated. So unmyelinated versus myelinated tissue. And the myelinated axons, they either form tracts, like we said, are in the central nervous system, or they are going to form nerves. Gosh, my writing is terrible, but anyway, the spinal cord, you have dorsal horns and ventral horns that are gray matter, and the dorsal horns connects to the dorsal root, and the dorsal root is usually sensory. And when we talk about it in humans, we call that the posterior, so it's your back. And the ventral root is like your belly, that is anterior, and it is motor. So it has motor neurons running through it. And so you have the ventral horns that connect to the ventral root, which is the motor neurons coming out the front, the anterior root. And so this is, the dorsal horns are going to receive the information into the spinal cord, and the ventral horns are going to take the efferent signal out. And again, I, have discussed this difference and discussed the kind of the reflex arc, and how you have some sort of stimulus, and then there's a receptor that receives that, that detects the change in the environment, and you have then an afferent signal that goes to the central nervous system. You then have some processing that goes on in the central nervous system, whether it's the spinal cord or the brain, and then you have an efferent signal going away. So you really should know these two terms, afferent and efferent. And then this just shows kind of the parts of that. So it's more pointed. This is the posterior or dorsal horn, so that is posterior. So anterior means ventral root. And so as the signal comes in through, and there's the dorsal root, this is the ganglia. I don't know why they didn't identify that particular part, but that is the ganglia. And then it comes in here, comes in and goes to the dorsal root, and then here there is some sort of connection, and then so again, this is coming in, that's the afferent signal, and then the anterior root's coming out. So that's how, and there's usually, with just a really simple reflex arc, they're just going to have that one signal, one neuron going in and one neuron coming out. But many times there's interneurons in between that, but just gives you the idea. Going in, receiving sensory information, and going out is going to have motor nerves, and it's going to send a signal to a particular brain, or sorry, a different organ that is going to make that particular function happen. So that's the spinal cord. And then you have the brain, parts of the brain. You can see this nice cross section that shows the gray matter, and it's very, well, fleshy colored here, kind of a peachy color. But the white matter is very white, and it's usually very shiny because of the lipids in the myelin sheath. It makes it look kind of more shiny, although the whole thing kind of looks shiny because it's wet with some sort of fluid on it. But this is just a cross section of a brain that they have taken out of a cadaver, and you can see all the folds that are occurring here. So these are the little convolutions that are going to give you more surface area. And then the other thing I would point out are these little holes that are the ventricles. So this is like the first and the second ventricle, and this would be the third ventricle. And then it comes down to a thing called the fourth ventricle down here that's between the pons and the medulla, or the pons and the cerebellum. And those are just the fluid-filled spaces of the brain. So what's interesting is that these convolutions become deeper and more separated, and the ventricles become bigger in someone who has Alzheimer's. So we'll show you kind of some pictures of that later when we get into that discussion. So the protective membranes around the CNS are the meninges. And this is important because there's a really very infectious disease called meningitis that affects people that are usually between 12 and 24, 25 years old. And it is basically an infection that you breathe in the bacteria. It comes through mucous membranes in the nose and the nasal area, goes through the sinuses and gets into the brain tissue somehow. And the three layers of this are the dura mater, the arachnoid mater, and the pia mater. Now, arachnoid means like spider, and the reason why they call that the arachnoid mater is because it has numerous connections, so it's kind of a spider webby kind of space there, the subarachnoid space. And that's actually where the cerebrospinal fluid will flow through this subarachnoid space. And then the pia mater is on the surface of the brain, and the dura mater is the outer layer, and in the brain it lines the cranium to protect the brain. So it lines the skull. And so the cerebrospinal fluid is there between the arachnoid and the pia mater in the subarachnoid space, and it absorbs shock. And, you know, when people get like a concussion, that's basically where the brain has been pushed against the skull so hard with a force that it basically bruises the brain. That's what a concussion is. But normally you absorb the shocks that happen just normally from doing day-to-day things, and your brain is really floating. Your brain would truly be so heavy if it didn't have the cerebrospinal fluid inside in those little ventricles we showed you and outside the brain, that it would crush itself. and obviously that would be bad. So you got to have these ventricles here inside, and then the cerebrospinal fluid is flowing around the outside as well. So you can see this, there's the first and the second ventricles on the other side, the other cerebral hemisphere. So this is like 1 and 2, and this is the third, and here's the fourth ventricle between the cerebellum and the pons. And then you can see it goes down the center of the spinal cord, and then it goes around the spinal cord and around the brain. So it's on the outside and it's on the inside, and everything is very cushioned and protected. It's much like, you know, when they put potato chips in potato chip bags, they'll put like air to cushion and keep the chips from breaking. And so a lot of times you have half of your bag is full of air and the other half is full of chips. So, you know, that's, you know, somewhat disconcerting when you really want more chips and you don't really get as many chips as you want. But anyway.